Morning, everyone, and thank you for joining us today in this session where we'll be talking about how to achieve ultra-high sensitivity detection of COVID-19 with a MIP-enabled sensor. My name is Francesco Canfarotta, and I'm head of chemistry at MIP Diagnostics. I'm also joined by Dr Marloes Peeters, who will give a brief introduction about himself whilst I set up my slides.
Thank you very much, Francesco. I'm Marloes Peeters. I'm a senior lecturer at Newcastle University in the UK, and I'm also acting director of chemical engineering at the moment. So I started working with MIPs during my PhD, and I've been working several years with Francesco to develop these MIPs into suitable sensor formats.
Thank you, Marloes. Okay, so we'll start by giving a brief introduction to MIP Diagnostics first. Then we'll discuss what nanoMIPs are and why they're used in sensor applications. And then we're going to delve into the COVID-19 nanoMIPs. In the second part of the presentation, I'll give the floor to Dr. Marloes Peeters, who will introduce her research group at Newcastle University and discuss the latest results using our COVID-19 nanoMIPs in the thermal sensor platform they've developed. At the end, we'll have a Q&A session. So should you have any questions during the presentation, just feel free to pop your questions into the chat box, and we'll answer your question at the end of the presentation. A little bit about MIP Diagnostics first. So we specialize in the development and manufacture of molecularly printed polymers. The company was founded in 2015 as a spin-out from the University of Leicester. We initially focused on custom MIP development. Then in 2019, we reached a £1 million revenue, and we secured our first MIP licensing contract. Last year, we secured a 5.1 million funding to expand our business and acquire new lab space and offices. Then they moved to perfect. This year, we became ISO 9001 certified and launched off-the-shelf MIPs. The industries we service at the moment cover biosensor and in vitro diagnostics, as well as pharma and oil and gas. Now, before we delve into the nanoMIP technology itself, we wanted to make this session a little bit more interactive. We would like to ask you if you have ever worked with MIPs before. If you can pop your answers in the chat box, even just a yes or no. Yeah, perfect. Wonderful. Okay, we've got some mixed. Okay, probably the majority has never worked with MIPs. Yeah, okay, most of you have not worked with MIPs. Perfect. Okay, so let's discuss about the MIP technology itself. As you know, MIP stands for molecularly imprinted polymers. Now, MIP can be manufactured in different formats. Here are the three main formats we work with, so bulk, nanoMIP, and linear. NanoMIPs are considered a synthetic alternative to antibodies with unique robustness and functionality characteristics. The three different formats allows a different usage. So, for example, bulk MIPs are typically used for purification thanks to their high binding capacity. And they essentially are made of large polymer particles between 30 to 100 micrometers. They possess selectable affinity, usually in the low micromolar region. Linear MIPs are conceptually similar to aptamers, so they are linear polymers of around 1 to 3 kilodalton in weight, with lower affinity compared to nanoMIPs. NanoMIPs are our patented core technology, which consists of discrete polymer containing one binding site per particle. We manufacture them usually around between 30 to 150 nanometers. They possess higher affinity compared to the other two formats, usually in the low nanomolar region, single digit, or even sub-nanomolar, and they're conceptually similar to monoclonal antibodies. As you may have noticed in the previous slide, we use molecular modeling to increase our chances of producing selective and high affinity nanoMIPs. We'll see how modeling works shortly. We'll now discuss more in detail how we produce our nanoMIPs. So we can divide this process into different steps. Initially, we immobilize our target molecule onto a solid phase, which usually is made of glass beads. The target molecule can be almost any sort of molecule, from a small molecule to peptides or protein. In this case, the template is depicted as a protein, and this molecule gets therefore covalently immobilized on the solid phase. In the second step, we add the monomeric mixtures, again, that may come from information obtained by molecular modeling. So monomers will self-assemble around the template molecule according to their functionalities. We then trigger polymerization. In this case, is depicted as a UV triggered polymerization, which leads to the formation of nanoparticles. Of course, some of them will be free in solution, not interacting with the template molecule. Others will be created around the template molecule. We then perform what we call a temperature-based affinity separation step. So essentially, we wash the solid phase with with solvent at room temperature initially to remove low affinity nano-MIPs, unreacted monomers, and polymeric chains that were not interacting with the template molecule. And then we increase the temperature of the running solvent in order to selectively collect the nano-MIP, the high affinity nano-MIP only. So as mentioned before, molecular modeling helps us in identifying the monomers that are predicted to have the highest affinity with the template molecule. The process can be divided in 3 steps. The screening, so first we code our template molecule, which can be a small molecule or a peptide within a protein, into our software. We then screen the template molecule against our proprietary database of monomers. The result will be a ranking table with the monomers ranked in a way that essentially allows us to use the information from the top part of the table, since these are the monomers that are considered to be good binders towards the template molecule. We can further refine this by looking at how and where the monomers interact with the template molecule. For example, we can look at where the interaction between monomer and a peptide molecule takes place, and we can look at which amino acid gets targeted, and therefore we can divide the peptide into regions and mix the different monomers that target different regions together in order to maximize on selectivity and affinity. The modeling works by looking not only at the functionalities, but also the hydrophobicity of the template molecules. We can also mimic the immobilization point, which is crucial for our solid phase chemistry. So we can almost hinder a portion of the template molecule, such that monomers cannot interact with that part of the molecule due to the fact that the molecule is immobilized onto the solid phase. And we can also mimic the actual peptide conformation within the protein. What are the benefits of nano-MIPs? So as already mentioned, nano-MIPs can be imprinted almost against any target. They can be easily functionalized, so we can introduce specific functional groups for coupling, for example, for conjugation or surface immobilization. They are exceptionally robust. They're proven to perform at extremes of temperature, pressures, and pH. They have selectable affinity depending on the end application. Since they're completely animal-free and they're synthetically produced, there is complete security of supply. And another characteristic, perhaps one of the most important ones, is the rapid development, since they can be produced in as little as 12 weeks, 8 to 12 weeks, depending, of course, on the complexity of the target molecule and the characteristics required in the final MIP. So why nano-MIPs are ideal for sensor? First of all, because of their ease of use. So unlike biological receptors, nano-MIPs do not denature during the coupling process. They can withstand extreme conditions, so they can be used to functionalize screen-printed electrodes for sensor development and be tested under different conditions. And as mentioned before, can be made against a variety of targets. They're not limited, so the nano development is not limited to targets that generate an immune response. So we can imprint also toxic compounds, and molecules of any size, from small organic molecules to peptide proteins, and even whole viruses or bacteriophages. On the right-hand side, you can see an image of the nanoMIPs that we produce using our solid phase approach. So now we're going to discuss a bit more in detail about the COVID-19 nanoMIP we developed for sensor applications. So we used a region of the RBD protein, so the RBD region of the spike protein for imprinting. This is because we wanted to essentially maximize the selectivity against SARS-CoV-2. We developed these MIPs in just 8 weeks. And then we subject our, these MIPs are subject to stringent quality control to ensure lot-to-lot consistency. Now, in here you can see, for example, a video of the nanoMIPs that we produced. This video was recorded on a nanosite, an S300. You can see the particles moving under flow and the random motions of the particles as well. The COVID-19 nanoMIPs were then characterized by SPR, and we assessed how the affinity of this nanoMIP is comparable to commercially available antibodies. We then proved that pre- and post-autoclaved MIP remained within specification of a KD below 18 nanomolar. And as you can see from this table here, both diameter and affinity is very consistent among 3 different batches, which again proves the lot-to-lot consistency. On the right-hand side, here you can see a dot plot analysis that was used to prove the selectivity of our nanoMIPs against other human coronaviruses. So you've got a nitrocellular strip onto which the different proteins were spotted. So you've got a row for spike protein and then other 3 rows for the other 3 coronaviruses, and the membrane was then incubated with labeled nanoMIPs. And as you can see, these 2 blue spots are the labeled nanoMIPs that binds only to the spots where spike protein was spotted. I'll now give the floor to Dr. Marloes Peeters, who is going to introduce her thermal sensor and show us the latest results with the COVID nanoMIPs.
I'm first going to introduce the research in my group and also show how this is a really good collaboration between academia and industry. Now, we work on different sensors in my group, and it might be interesting to hear from you what kind of sensors that you might be using. So again, in the chat, maybe you're working with electrical sensing, maybe you're working with optical sensing, or you use different methods. So please pop in the chat what you're using in the moment, and then we will discuss a couple of different options. Now, in my group, we look at, for instance, thermal sensing, where we looked at we have a portable device that we look for contamination in water and in food samples. So this could be, for instance, here you see a picture of E. coli, so of different bacteria. It could be, again, where we look at viruses, where we also started looking at norovirus, but also potentially for biomarkers, such as cardiac biomarkers. And we also had a poster here where we looked at cardiac biomarker sensing. Now, for electrical sensing, you might be thinking like, okay, I want to detect biomarkers in vivo. And obviously, you can't just apply a temperature pulse in the body. So electrical sensing is a really interesting alternative, and we will show you that this is also feasible with the nanoMIPs if you integrate, for instance, a redox probe. And here are some of the projects that we worked on, where we developed a new catheter which can actually measure inside the bowels. So looking at neurotransmitters there, and we've also been working at a cochlear implant to monitor inflammation. So there's several different options, but for today, I'll mainly focus on our thermal sensor. Now, you get these nanoMIPs, and then the first question that you might have, or the first complication, is how are we going to get them onto a surface? And there are many different ways of doing this. So obviously, they usually come like just in water in a solution. So if you don't want to worry too much about the methodology and want to keep it simple, the first thing you can just do is either use drop casting or dip coating. And we have shown that this is possible. And we've seen quite good coverage if you have electrodes that have, like, for instance, like a rough surface, like these screen printed electrodes, for instance. And then they are physically absorbed, and they are relatively stable if your surface is not too smooth. But what we also know then, and that what we do see, is that even though the surface coverage might be quite good, obviously you might be worried about reproducibility. And also the nanoMIPs might not necessarily be in the best orientation in order to facilitate the binding. So what we've done in our latest paper is that we really compared a couple of these different methods and shown that if you do use a covalent coupling method, then this is much better. And there are different ways of how you can do that, because these nanoMIPs have a lot of functional groups. So if you use a typical like organic coupling, like for instance EDC coupling, this is very much possible. But what we've done is relatively fast. So we first do an electrografting step with a diazonium salt, and then we do EDC NHS coupling with the nanoMIPs. And we have seen that this leads to really good surface coverage, and it can be worked across like a number of different surfaces. So whether you're working, for instance, with a gold surface, a silicon surface, or what we have shown that's very interesting is working with these screen printed electrodes, which are very low cost and they're very reproducible, but very interesting from a commercial perspective. Now, even more so, here I've shown that this is done in 2 distinct steps. It is also possible, and we only kind of recently started exploring this, is to do it as all in a one pot reaction. So to do it even faster than that. The other thing that you might be interested in is that Francesco has shown that these nano-MIPs are very stable in solution and are stable to like extremes of pH and temperature. But I presume you also want to know that they're stable on the surface. And what we have shown is that using AFM, and then we actually look at the size, so not so much at the affinity, is that we started heating it up, and we also exposed it to different conditions, including different pH. As you can see, there's no change in the size as well, which is also an indication that the affinity is retained under extreme conditions. So if you would be, for instance, wanting to use a COVID sensor, let's say, for instance, in wastewater or something where, you might not necessarily have room temperature. Now, when we then go to this kind of thermal sensing that we work with, during my PhD, I started working on what we call a pore blocking model. And this is where we discovered that if you think of MIPs, which are porous materials, or they contain these binding sites, once you get binding into these binding sites, you're blocking the surface. So we can see, and we already knew that it blocked the electrical resistance. So if you imagine and it would always find the path of the least resistance, all of a sudden you get this binding of these organic molecules into the MIP. So you can see that you really block the electrical resistance. But what we then show, which is quite different, is that it also started to increase the thermal resistance. So if not it's just an electrical pulse, but if you would pulse heat through the surface and then you're kind of blocking the pathway of the heat, we can also see that there's an increase in thermal resistance. And we've really shown that this is applicable to like a wide range of targets, starting off with neurotransmitters. But by the versatility of the MIPs, we also apply this to bacteria and, for instance, other targets. Now, what is, then that advantage of this like thermal sensing over electrical sensing? In a way, what you're doing here is it's not more than just measuring 2 different temperatures. So when you're doing this electrical resistance, you might need to do impedance spectroscopy. So you might need to work with like complicated data, a lot of data post-processing, and maybe expensive equipment that you don't have access to. So it's really low cost and an easy way of doing this. And on the right, you can see that you do see this increase in electrical resistance when the target binds. Whereas if we look at a non-imprinted polymer of a binding of something else which is similar to the target, we hardly see any increase in this thermal resistance. So if you want to find out more, I have put some of these references below there. But we also have a YouTube channel where we showcase some of the work, particularly on antimicrobial resistance. So you can also have a look there to see what our lab actually looks like and how we use some of these nanoparticles. Now, what does this device look like? So I'll first show you like a little picture of what it looks like, and also show you some of the changes that we've made. The device itself is there in that closed off box. So this is what actually measures the temperature. And in the video that I'll have on the next slide, I'll actually go a little bit deeper and show to you what it looks like from the inside. Now, we have shown here a syringe pump because you can automate your measurements. But also what we're starting to do now is we have a type of setup where you just pipette in your solution. And the kind of quantity we're looking at is really quite small. So it's a sample volume of around 100 microliter in that particular flow cell. And to give you an indication, that's about 3 drops of blood. So it's really quite a small sample volume that you need. So within that flow cell, this is where you actually put the electrodes in that are functionalized with your nanoparticles or your MIPs. And then you have that device that would measure the temperature for you. Now, if you are interested in what this looks like from the inside, this is a very short video, so hopefully it's quite self-explanatory. But here you can see this is opened up, and it will show you kind of how this works, and it will also hopefully give you a feeling for how these electrodes work. This is the electrical signal, which can give out a certain voltage, so you can increase or lower the temperature. And then beside that, you've got resistance, which you can use between the voltages. And on the outside, you've got the inlet with thermocouples that measure the temperature where they go in. And here you can see these little cells into which we fit the electrodes. As you can see, they're exactly the same size and shape of the electrodes. These flow cells are made using 3D printing. The 3D printing device that we have at home melts polymers in order to make them in any size and shape. Now, what we do in the end to close off the system, because you don't want the water to go anywhere, so we add a copper block on top. Copper is very conductive, so you can use that to monitor the temperature that goes from the copper back to the device. Okay, so this is a little insight into how this actually works. So this was shot in my lab. But now the more interesting thing is obviously the results, and also comparing this to, for instance, antibodies, and looking at that kind of selectivity that was demonstrated by Francesco too. Now, what we can see, and we've tested this, and on the left-hand side, you can see that this is increase in thermal resistance, and this is what we expect. So once we get binding of the target of interest, and we look both at the full spike antigen and the receptor binding domain, you can see that there's a gradual increase in thermal resistance as you start to increase the concentration. And this is what we would expect, because you've got binding to that layer, so you start blocking the heat flow, so you would see an increase in thermal resistance. And what we then did is we compared this to antibodies. So we did exactly the same experiments with antibodies, and also compared the limit of detection that we got. Now, thinking of, you had that spike protein, and both for the receptor binding domain, we were able to get between, let's say, around 10 femtograms per milliliter. If we would do it with the antibodies, and you can also go to the website of MIP Diagnostics, where they have a technical data sheet where you can download some of these graphs, we found there was a 20-fold increase compared to antibodies using exactly the same functionalization method and the same detection technique. So we can show that they do kind of outperform antibodies in this particular sense. Then we also started looking at the selectivity, which we've tested with a variety of different compounds. I'm not showing all of them, but we looked at, for instance, one of the open reading frames, where you can see there's hardly any response. We looked at BSA, we looked at HSA as well, and a number of different antigens. And while in the other one you can clearly see an increase in the thermal resistance, this is not present for some of the other compounds. So this clearly indicates that this sensor is also selective as well. If you are wondering how that kind of limit of detection then compares to, for instance, lateral flow tests, and it would be interesting to hear from you how much you trust the results that you get in lateral flow tests. Here in the UK, we can order these lateral flow tests to get them at home via Innova, and there's been a lot of commotion around the topic that they are perhaps not quite as sensitive as we want them to be. But besides this, if you compare like this 10 femtograms per milliliter, this is comparable to what you can find in literature using other different approaches, and a lot of you might have seen other presentations on the topic. But it is definitely much more sensitive, and at least by a factor of 1,000, if not more, compared to what you've got with lateral flow tests. So meaning that this is a really nice and quick, interesting alternative. Now, what do we need to do? And here I also wanted to show a picture of my research group, particularly Jake McClements on the left, who's done most of the experimental work that you see here. There is obviously a lot of different steps that we need to take. And we have seen that if we use an addition type of flow cell, where you don't do anything differently than just pipetting the sample in there, that we can really get down to very fast measurements time. Because even with a lateral flow test, now you need to wait at least 20 minutes, and that can be quite long, you know, thinking that you might want to go to like a concert or something and imagine everyone have to do a 20-minute test. And we have shown, for instance, with a similar technique that we used for a cardiac biomarker, troponin, that you can really go down to around 5 minutes. So it can go a lot faster. But besides that, all the kind of like studies I've shown here were done in PBS buffered solutions. But obviously we would need to work with clinical samples. And we are in the process of measuring blood and saliva samples, and really comparing those results, directly benchmarking them against existing tests and lateral flow tests. In Newcastle, we also have class 3 lab facilities, which also means that we can test with the live virus in-house. This would mean, and we have had discussions around that because this setup is completely portable, it doesn't weigh much, you can just take it with you, you can plug it in, and you can directly use it wherever you want. So what that means is that we can give the clinicians that actually have the experience of working with this virus, we give them our little box, they're trained in how to use it. So it's not that difficult to use. They take it with them, they plug it in, and they can actually measure for us there. The other thing that you are probably interested in, because you might not be interested in COVID-19 sensing alone, you might be interested in looking at, for instance, other biomarkers. And one of the beauties of working with these nanoparticles is that you can transform it into array formats, and you can target a range of different applications. So whereas you might be able to use like an array format for an ELISA where you've got compounds of a similar size, here you can really look at, for instance, targeting both bacteria and viruses and smaller particles at the same time. So the versatility of these nanoMIPs is a real strength. What we're also considering at the same time, and recently we did a little bit of work on optical detection as well, where we conjugated or we incorporated some fluorescent probes, so we can have a dual detection platform. And that would still be really interesting, because if you directly want to validate your technique, ideally you want to do both at the same time, so you can really trust your results and see that they are reliable. And we are now in the process of using, for instance, like you can use some monomers that are electrically active. You can use some ferrocene probes. And if you incorporate them, and this platform is also simultaneously able to measure electrochemical impedance spectroscopy, you can directly combine the both. Because if you think of it, if your thermal resistance is changing, we know for sure that there's also an impact on the electrical resistance. And even more so, like we can take this setup, and we're using this at the microscope at the same time. We can use this fluorescence microscopy at the same time too. Now, hopefully this gave you like a bit of a whistle-stop tour on like what are some of the advantages and what are some of the work we've done on COVID-19. And it would also be interesting to hear what do you think about sensors, and if you're potentially thinking of commercializing your sensor setup. What we also wanted to bring to your attention is that Francesco and myself are having a special issue in chemosensors. So this is on the advances and manufacture of sensors based on MIPs. So we've seen here that some of you are working with MIPs, some maybe not, but we can see that for a lot of you this might appeal to. So we are looking for papers that are looking at MIPs and particularly at sensor aspects. So if you'd like to know more about this, obviously you can contact us.